is our first lecturer in this series. Professor Fernandez pioneered the research in the mechanical aspect of protein folding dynamics. He's giving now a very inspiring course at Tel Aviv University while he's visiting us as a Sackler scholar. And we are looking forward to hear you. Please, Julio. Thank you.
So thank you, Ori. It's a real pleasure to be here, and I'm here on sabbatical. And one of the nice things of being on sabbatical is that you're away, and that the students don't try and get in touch with you because they know that there's not going to be much feedback and so on. And I replaced all of that with a wonderful new set of students that I have here at Tel Aviv University, where I brought one of my setups. And I'm teaching an experimental class on single molecule force spectroscopy. And there are many reasons why I'm here. I really like coming to Israel, and I think that the Nano Center has been a great friend, and I'm grateful for their invitation. But also from the intellectual scientific point of view, it's a real pleasure to have active, excited students. And those are rare to find today. So you're trying to think on how Tau competes with first-year universities in the United States. And I think that you'll find that some of your students here compete with anyone in the world. And so it's not just a matter of equipment, but I think it's mostly human capital that really makes a big difference. Having said that, I still have not graded my students, and so we'll find out at the end. But they're really all exceptional people, and it's a real pleasure to be teaching here. So the research in my laboratory is focused on trying to understand the effect of mechanical forces on the dynamics of proteins. And you may wonder if that's just something very esoteric that's of interest to me, and not necessarily to you, but you will be very wrong. Because in fact, most things in biology are mechanical, and from the simple things of pinching your skin and the skin being able to go back to its original shape, or doing this and your brain remaining intact, well, in most cases, and things like that. So really, when you think about the effect of mechanical forces on molecules and biology, we know so little about it, yet it's so prevalent and so important. And the reason is not just because I had an inspiration and I said, wow, let's study the effect of force on molecules, but rather the techniques really truly became available only with the advent of the atomic force microscope in the 80s, and also with molecular biological techniques that let you engineer proteins at will, which are very easy to use today. But many of you might be familiar with ion channels. Ion channels are always present in the membrane of most excitable cells, and ion channels open and close and let ions through, and ion channels are mechanically gated proteins. They sit in a very intense electric field, and when the electric field is switched on during an action potential, the ion channel opens and closes as a function of time. That electric field activates some charges in the structure of the protein, and the charges exert a mechanical force on the molecule, creating the opening that allows the ions through. The tissues, all of the tissues in biological systems are held together by something called the extracellular matrix, which is a mixture of polysaccharides and proteins, and the cells interact with the extracellular matrix by applying force and sensing the elasticity of the substrate. And again, in that case, the mechanical response of those molecules is not just to serve as a scaffold, but also in a tight interaction with the cells. Synaptic transmission, if you want to think about something more prevalent and common in neurobiology, synaptic transmission, the connection between a neuron and the postsynaptic, in this case, the neuromuscular junction. Again, the distance between the neuron where this neurotransmitter is going to be released and the postsynaptic membrane where the action potential is going to be triggered is rather critical, and so it's important that they stay in register, and you have a number of molecules that mechanically hold these structures together in a very precise fashion. Muscle, of course, is sort of the ultimate example for mechanical tissues. There's a huge number of molecules in muscle that are in charge of generating forces, but also in charge of the elastic properties of muscle. One has to think that in the human body there are about 600 different types of muscle, and each muscle is particularly well suited and tailored for the function of that muscle. So it's not that there is just one set of proteins for all of them, but rather there are alternatively spliced proteins that are tailor-made for the elasticity and the contractility of each individual muscle in the human body. Muscle, as you may recall, is made of individual contractile units called sarcomeres, and here is an exploded view of a muscle that I took from Scientific American. You can see here an entire muscle, you can see the nerves here, the neuromuscular junction, and then going all the way down to a single myofibril with the contractile unit, which is the single sarcomere. You can almost see here the C lines. And the sarcomere, as a contractile unit, can be visualized rather easily in a regular microscope. A single myofibril from a muscle fiber can be isolated, and you can look at it under the microscope. And here is a single myofibril that's glued to a pipette here. You can see some of the glue on the left, and glued to another pipette on the right. And the idea is just to stretch it so that you see what happens to the individual sarcomeres. And you see here that the thick part of the sarcomere doesn't change. That's where all the contractile machinery is. But the so-called I-band of the sarcomere, where the elastic proteins are, extends and then contracts and extends and contracts. And this is all just the passive elasticity of a sarcomere. And so it's quite amazing that one can take tissue, such as muscle tissue in this case, stretch it a lot, right? And then you relax the force, and it comes back exactly to the configuration that you had before. And evidently, you have to be able to do that an infinite number of times so that your muscles will be active throughout the life of the organism. And all of this made of proteins. Proteins made, different types of proteins assemble into a single sarcomere to allow this action, to allow the contraction of the muscle, and to allow also for the elastic extension of the muscle. And I'm going to go very quickly through this, but one of my favorite proteins actually comes from muscle. It's called titin. Titin is the largest protein we know, and titin is responsible for the elasticity of muscle. So when you stretch a muscle myofibril passively, what you're stretching and extending is titin, which is extending in response to a mechanical force. And so that these things are very common. The way proteins respond to mechanical forces is an essential component of biology, and it has been very difficult to study before because most of the studies of proteins were done in test tubes. You purify the protein from the tissue, put it in a test tube, and then measure some properties such as the change in fluorescence from a probe or something. First of all, the protein is isolated from the tissue. Second, you could never apply mechanical forces to the proteins in this case, so you would just be looking at relaxed proteins. And so that made it very hard to really understand the dynamic and properties of elastic proteins such as titin from muscle. And there are many, many, many examples. I could go on for a day listing the kinds of proteins for which it's essential to know really how they respond to a mechanical force. So titin is just one of them. Titin had been studied this way. They had taken pieces of titin, which is a very large protein, cut it into smaller pieces, about 100 amino acids long, and then studied the properties using chemical denaturants, which is the standard way that most protein biochemists have used to study proteins. And so for most of you, this is like totally alien territory in the sense that protein biochemistry is something very distant. But I remind you, proteins are perfect nanomachines. A single protein of the ones that compose titin will be about 5 nanometers in length, and it has this amazing property of being able to extend on the force and then refold exactly in the same conformation an infinite number of times, where every atom goes back to roughly its position spontaneously without requiring any additional set of reactions. So it's pretty important to develop techniques to try and really understand how proteins in general, from every tissue, from every question that you can come up with, respond to mechanical force. And that could never be done with the standard tools of protein biochemistry. So this is really sort of landed squarely on our lap in terms of using nanotechnology to be able to understand one very, very important part of biology, which has really not been explored before. So the way we do it in our laboratory, and I began this about 10 years ago, and it was sort of a combination of luck and accidental experimentation together with Hermann Gau. It was another sabbatical. I was in Munich, and Hermann was an expert in using the FM, and I wanted to study proteins, so we got together and we did some simple experiments, and we were able to see that we could actually pick up a single protein, apply force to that protein, and then really look at the dynamics of a single protein, which was pretty amazing to us at the time. And so the way this was done first, and it's done today in my laboratory, is that we have an atomic force microscope that's simplified here. You have a cantilever, you have a laser beam that deflects off the cantilever into a pair of photodiodes, and that deflection is a measure of the force. And then you have a piezoelectric actuator that extends the protein, so you can adjust the length of the molecule. And this is done under feedback so that the piezoelectric actuator would extend to whatever is needed to keep the force to the set value. So it's an approach that we call force clamp spectroscopy. And it's a form of spectroscopy because we tilt the energy landscape of the molecule by applying a mechanical force and doing work on the molecule. So that's why we call it single protein force spectroscopy, but it's really force clamp spectroscopy. And we buy some parts, and this is where the interdisciplinary part comes in. We make our own proteins in the laboratory, and we write our own programs, mostly in Igor, but we also build our instrumentation, most of it. And we buy parts from FM companies, and we modify them so that we can actually do our experiments. So you'll see here, it's a part of a Pico head, which the nanocenter's got several, and modified to have a decent laser that has to be a sort of high-quality focus laser. And here we get top-of-the-line piezoelectric actuators from Physik Instrumente and so on. We change things to optimize the system, not for profit, but for its ability to detect a single molecule. A typical protein we use, one example is shown here. This is a protein from Titan. Like I said before, it's about 5 nanometers in length by 3 nanometers. It's a typical protein in the genome, proteome. It's about 90 amino acids. It's the most common size. And in this particular case, it's called an immunoglobulin beta sandwich. And picking up a single protein is difficult, so we use molecular biology to construct proteins which are made specially for the FM experiments. And it's a bit of a cultural shock to hear that, but you can actually use protein biochemistry to construct molecules that are specific for the kind of instrument that you're using. And so in our case, we make the so-called polyproteins, where we take a single protein, the beta sandwich I just showed you, and then we repeat it many times. So this is a repeat protein where the repeats are all identical to each other, and it's constructed at the DNA level, and then we express it in bacteria, we purify the protein, and we end up with a relatively pure sample of polyproteins, which are the proteins that we pick up with the atomic force microscope. The result of picking up a polyprotein under these force feedback conditions is a staircase of identical steps. Typically, in the proteins that my students have been playing around in the lab, these steps are 20 nanometers, and correspond to the extension of the polypeptide when a protein unfolds. When a protein unfolds, the backbone extends under force, and you get these stepwise increases in length of the protein as the feedback pulls on the protein in order to maintain the force at a constant value. Each one of these steps then corresponds to the unfolding of one protein in the chain of the polyprotein. And therefore, if you get many events, you can actually measure a rate of unfolding at the particular force that you're using. And that's sort of the essence of the measurement. If you have to have some sort of a picture in your mind of what this looks like, this is a scanning electron micrograph of a real cantilever. And the scale here is approximately 10 nanometers here. So you see the tip is maybe 20, 30 nanometers, depending on the state of the tip of the FM cantilever. These are actual structures of the protein that we use, not the entire thing, but the individual components here of this polyprotein are the correct PDB structure obtained from the protein data bank. And more or less it's all to scale. So you can see more or less what this must look like when we pick up a protein and stretch it. It's probably something more or less of that scale and those dimensions. So if you do that many, many times, and the AFM that we construct is automatic. So it goes down, picks up a molecule, stretches it, and it repeats that many thousands of times during an experiment. And on a relatively low number of cases, somewhere between 1 and 10% of the cases, you pick up a molecule. And because it runs on automatic, after a day of experimentation, you get many of these traces. And these traces, as you can see, have one feature that's common to all of them, and are this 20 nanometer step increases in length caused by the unfolding of a single protein. However, the times at which these events take place vary a lot from trace to trace, because the unfolding is actually a probabilistic event. It's not something that's deterministic. There isn't an event that's going to happen at a particular time, but rather it's just a statistical mechanical property of the protein to unfold. But if you sum many of these traces and average them, you get a smaller trace that you can see here. And that smaller trace, now you can fit a single exponential to it and then obtain the rate of unfolding at that particular force. So it's a rather straightforward procedure. Pull polyproteins at a constant force, obtain a few traces, sum them up, average them, and then fit them with a single exponential, and you obtain the rate of that particular reaction. This is the essence of what we do. Once you can measure rate, there's an infinite number of questions and problems in biology that you can address. Not just what I'm going to show you, which are just a couple of examples, but really there is an enormous variety of questions on what this particular mutation would do, what happens if we phosphorylate this, or, it's just a sort of an unending theme. So for us, the key is sort of to develop the groundwork for this and try and develop some of the questions that we have approached. So the simplest thing that you can do with this measurement of the rate is to measure the force dependency of the reaction. So you measure the rate at different pulling forces, and in this case, this is for protein unfolding, as I showed before. You measure protein unfolding at 120 piconewtons, at 140, 160, 180, and so on. And you measure the rate using those exponential fits, and you plot the rate as a function of the pulling force. And this is a semi-logarithmic plot, and you can see that the relationship is that of an exponential. And this is very ancient. Svante Arrhenius already had come up with a way to explain the temperature dependency of chemical reactions long, long time ago. And he said that the rate will be proportional to sort of a prefactor constant multiplied by an exponential term that includes the activation energy barrier divided by the temperature. And the equations that we use are all modifications of Svante Arrhenius formulation. And you can see here that we've added now a term, an exponential term, where the rate now depends exponentially on the applied force. And this factor here, the distance to transition state. And the logic of this is that when you apply force, you do mechanical work on the energy landscape of the protein. And the transition state of any reaction is the most energetically costly structure that you have to traverse in order to go to the other side. It's like crossing a mountain pass, and it's typically called the saddle point of the reaction. So everywhere else is high energy, and the saddle point is the structure that you have to go through to go from reactants to products. And the distance is a real distance, it's a real molecular distance from the native state of the molecule, in this case a protein that's unfolding, to this transition state. And when you do mechanical work, you reduce the height of the activation energy barrier by an amount equal to the force multiplied by that distance. Force times distance is work, and so the amount of the reaction in the energy barrier that you have to cross equals that term, force times distance to a transition state. What was immediately interesting to us is that when we measured the force dependency and we applied this simple Arrhenius formulation, that distance to transition state turned out to be pretty much of the size of a single water molecule. And that was a lovely result because intuitively you could imagine that a protein, be that in the elastic tissue of your heart or anywhere else, something has to happen in the structure for the protein to unravel and extend. And crossing that energy barrier clearly will involve some solvent molecules. And so the moment that you break the protein open and you allow the solvent molecules to come in, the protein will extend. And so it was an interesting assumption that we made that this number really corresponds to that of a water molecule. And the saddle point and the interpretation, the molecular interpretation I just gave you, actually is not just things that we come up with, but other people have done beautiful experiments using, for example, molecular dynamic simulations. The work of Klaus Schulten at University of Illinois in Urbana-Champaign has shown that for a protein to go through the saddle point, you have to acquire an actual molecular structure that's really well simulated by molecular dynamic simulations. And I'm going to show you one here for a protein named ubiquitin. And the dashed lines here correspond to actual bonds that are breaking as the molecule is pulled. And using the computer, you can actually generate a complete history of the extension of the protein from the initial native state to the fully extended state with a history that goes bond by bond breaking throughout the entire structure. And one of the things they predicted for us was that indeed that the structure will contain solvent molecules. We could do the experiment to confirm this by measuring the force dependency of the unfolding of this protein from human heart titin as a function of replacing the solvent from water to glycerol. And the reason we like glycerol is that glycerol is a bigger molecule, so it occupies a bigger space. And if the solvent played a role in the structure of the unfolding transition state, by having a bigger solvent molecule, that distance to a transition state would increase. And indeed, that's what you see here as you replace water by glycerol, going from 0 glycerol to 100% glycerol. You can see that the distance to the transition state increases very substantially from the size of a water molecule to the size of a glycerol molecule. We have done this for a number of other solvents, so we can increase the size of the solvent. And what you find is that as you increase the size of the solvent, the distance to the transition state increases with the size of the solvent until the solvent gets too big
and those molecules no longer can get into the structure of the transition state. So this is something that we can measure quite well. The distance to the transition state is a real measurement of molecular distance rather than some theoretical invention. And we continue this question by looking at force-dependent chemical reactions, which takes us in the other direction. It takes us from molecular size to quantum mechanics. And for the longest time, man has used force to catalyze chemical reactions. I mean, mortars and pestles had been used for a long time to sort of mix things together and change them, right? But you could never establish exactly which direction the force was being applied. And with the FM today, we can actually define precisely the reaction coordinate where we apply force. And by applying force to a molecule, we can actually observe what the effect of the force is on a chemical reaction, quite different from the way this had been studied before. And so our essay is shown here. We study a reaction that's called the thiol-disulfide exchange, which consists of catalyzing the reduction of a disulfide bond, which forms between two cysteines that we have engineered into the protein. And you can see here the cysteine residues are labeled here in yellow. And we apply force to the protein. The protein extends and unfolds, but it can unfold only right up to the disulfide bond, because the disulfide bond is a covalent bond. It cannot be broken by force. And so if when you unfold the protein and you stretch it to the point where the disulfide bond is exposed to a solution, if you have a molecule in the bath that can actually reduce the disulfide bond through an SN2 type chemical reaction, then you get a second extension of the protein, which corresponds to the number of amino acids that are trapped behind the disulfide bond, and that would extend the protein further. So before, for the unfolding, we had a single unfolding step that corresponded to the extension of the full protein, but now we're interrupting that unfolding step by creating this disulfide bond that can only be forced to extend by a chemical reaction. So we can see then how force applied to this disulfide bond catalyzes that chemical reaction. I'll go back to this so that you see what I mean in detail. This is sort of an animation of what the essay looks like. Remember that we're under feedback. We're applying a constant force to the molecule. Here is a trace showing you the length of the protein as a function of time and the constant force. We're applying the force. The protein extends by unfolding. And you see a step increase here of 11 nanometers corresponding to the extension of the protein right up to the disulfide bond. The disulfide bond does break the protein because it doesn't allow the extension of the polypeptide any further. But if you have the nucleophile in the solution, the nucleophile comes in, reduces the disulfide bond, and you get a second step in the extension, which you can distinguish readily because the number of amino acids that you release upon reduction of the disulfide bond is different than the number of amino acids from the initial unfolding. So it's a difference between 11 nanometers and 15 nanometers. So we can tell, in this case, precisely what is unfolding and what is a reduction event. I'll run this again so you see the action here. So the protein is folded. We're applying a constant force. The folded length of this protein is around 4.5 nanometers or so. It unfolds. It extends to 15 nanometers. You measure the difference in the extension, around 11 nanometers in step increase. But then if you have the reducing chemicals in the bath, they'll come, reduce the disulfide bond, and you get a second extension. So the first one is simply the mechanical unfolding of the protein. The second one, the second extension, the second step is the result of a chemical reaction. So this actually is rather easy to do. here is an example of what I meant in the animation. You have a protein, a polyprotein, made of many repeats, all containing the disulfide bond. In the absence of the reducing agent, you just get the 11 nanometer steps corresponding to the unfolding of the protein, but you do not see anything afterwards. If you put the reducing agent, and in this case we're using around 12 millimolar, you get this first series of steps corresponding to the unfolding of the protein, but then you get a second wave of steps, bigger steps, 15 nanometer steps, corresponding to the reduction of the disulfide bond in the same proteins. And this is also done under force, because we apply a pulse of force to observe the force dependency of this particular chemical reaction. So I don't know how many of you do chemistry, or how many of you do single molecule work, but in here, think about it for a second. Each one of these steps is the result of the action of one nucleophile, one reducing molecule, DTT for example, and two sulfur atoms which are covalently bonded. And when the nucleophilic attack takes place, the disulfide bond breaks and the rest of that protein extends. I mean, this is just one chemical reaction in two atoms being able to be seen by a simple assay with atomic force microscopy. It doesn't get any more nano than this, I think, in terms of the scale of the events. As before, you take many number of these events, and because remember that you're measuring the rate of a chemical reaction at a single molecule level, so you count the number of steps of reduction that occur per second, and you can, as before, compile an average, fit it with a single exponential, and obtain a rate. And you can see that as you increase the applied force, the rate of reduction of the disulfide bonds increases as well. And you plot that as a function of force, and you get again that the rate increases exponentially with the force. And it's interesting because this is a chemical reaction. The rate increases linearly with the concentration of the nucleophile, which is DTT, but increases exponentially with the force, and you can see that here. And so it's the sort of hallmark of a simple bimolecular chemical reaction. You can see the description here. It depends on the concentration, and it also depends on the force. What's interesting is that if you plot the rate as a function of force and you measure the slope, you get that the distance to transition state in this case is less than 1 angstrom. It's a distance of 0.34 angstroms. This is way smaller than a single water molecule, and we've gone from a molecular structure, which is the transition state for unfolding, to really quantum mechanics. And the explanation for this value comes from people who have done density functional theory simulations of nucleophilic attack, and people have shown that the disulfide bond elongates by about 0.4 angstroms at the transition state of a nucleophilic attack, and this has been done, like I said, using DFT and quantum chemistry. And so our measurement matches quite well that of the predicted transition state. Now, you're probably wondering, well, Julio started talking about biology, and now he's talking about density functional theory. Are the two related in any way? Well, I think that the second question that's interesting is that we've studied all these things in test tubes and so on, but is the chemistry of this biochemistry that we hold so dearly changed by applying force? I mean, when you do this, is the chemistry in those molecules that are being mechanically stretched affected in any way that we just don't know about? And I have some gruesome examples on how to think about it. Let's say that you are in a car accident and you break bones. You're going to have to imagine that there is a lot of tissue damage caused by mechanical injury. And in those tissues, there's going to be some very large mechanical forces that are applied to the tissue. If the biochemistry that takes place around those tissues is force dependent, then the biochemistry that you study in a test tube is going to be very different. And so this is also a biological question. Is chemistry affected by mechanical forces? And as you can see here, the very simple chemistry of a disulfide bond being reduced by a nucleophile is force dependent. And for the sake of chemistry, some of my students have looked to see whether this distance to transition state that corresponds to the elongation of the bond at the transition state depends on the nucleophile, and different chemical agents, different reducing agents, have completely different slopes in terms of the distance to transition state. If you have a phosphorus atom being the central atom for the nucleophilic attack, the distance to transition state is longer. If you have sulfur and you have thiols, the distance to transition state is shorter. And there's no sort of density functional quantum mechanical calculations of this, but we're very hopeful and very arrogant to think that the chemists are going to be very interested in our results and start making simulations of chemistry under force. We also do some things that you always ask, well, have you measured the temperature dependency? And the first thing that you have to know is, well, you should be asking why should we measure the temperature dependency. Temperature dependency is actually a really sort of simple experiment, but it can give you very important numbers that are hard to get in any other way in terms of the energy landscape of the chemical reaction. So we've constructed another AFM, and you can see here that it doesn't look like an FM anymore. This is truly with no parts from Vico or Asylum on any of those companies, except for the fluid cell. Everything else, it's actually ordered from different companies. This is just an optical table, and horizontally we mounted this high-quality piezoelectric actuator. We have a stage for the diode laser here. We have our own photodiode detector. And you see here a close-up of the piezoelectric actuator, and with a Peltier element here, and a way to remove the heat of the Peltier so we can cool and heat also on the sample surface. So we have very good control of the temperature, while at the same time the instrument can look at this chemical reaction. And well, you can see that the thiol disulfide exchange chemical reaction, if we hold the force at a constant value of 250 piconewtons, the reaction is quite temperature dependent. And again, this is measured the same way. You measure the frequency with which you see reduction steps in the extending polyprotein at a constant force. And it's temperature dependent, and at constant temperature, of course, the reaction here is force dependent. So you make those two measurements. You make measurements of the force dependency of the chemical reaction at different temperatures, and you obtain these plots of rate as a function of force. I'm not going to go through all the details. You've seen this a million times. You can finally pull all the data together in a semi-log plot of the rate as a function of 1 over the temperature, and then you get this slope, and from this slope you can actually get a very precise measurement of the activation energy barrier of the reaction and of the prefactor of the reaction, which is the rate at which the substrate disulfide bond and the nucleophile are colliding with each other to produce the transition state of the chemical reaction. So this is all kind of biochemistry or chemistry at a single molecule level in the stage of an AFM instrument. And as my students will tell you, with some difficulty, this works every day. And so you can, in fact, you're all invited to see, I have Amir here, and you know, some of my students which are here in the group are able to operate this instrument pretty much on a daily basis. Not all the time. It requires some skill to get it going and produce good data, but But generally, you can see a single molecule experiment on demand. And so really that's what's essential for us to be able to do this kind of AFM chemistry rather than test tube chemistry for studying these reactions of molecules under force. This is going a little slower than I thought, so let me just jump over this and take you to the enzymes. Because one of the interesting questions that I asked before is, well, what happens to the chemistry of the tissues if you apply a mechanical force, let's say during mechanical injury, during circumstances that would expose tissues to very high pulling forces. And we don't have molecules such as DTT, we have glutathione, we have some reducing agents that reduce disulfide bonds, but we do have a lot of thioredoxin. Thioredoxin is an enzyme that catalyzes the reaction of disulfide bonds in just about all tissues, and is a very early ancient enzyme that is found in all organisms. And the feature that is constant to all of these thioredoxin enzymes is that they have two cysteine residues that are essential for the chemical reaction. One of them is called the catalytic cysteine, and is the one that does the reduction of the substrate disulfide bond, and the other one is responsible for maintaining the catalytic cysteine in the active form. Again, just to show you the scale, a single thioredoxin enzyme will be roughly about that size. That's the actual structure. And this is the polyprotein being extended. More or less, that's the scale of the experiment. And as before, we unfold the polyprotein, exposing the disulfide bonds to a solution, and then we look for a second wave of steps corresponding to the reduction events where the enzyme has catalyzed the reaction of the disulfide bond. And you can see here that now we're using micromolar concentrations of the enzyme as opposed to millimolar concentrations of the other types of nucleophiles, such as DTT, that we had to use before. And that's because enzymes are incredibly effective. That's the reason we're alive, is because enzymes catalyze chemical reactions, increasing the rate tens of thousands, hundreds of thousands, millions of times. And the exact form by which enzymes achieve such a tremendous increase in the rate of the chemical reactions they catalyze is not really well known. Part of the reason that it's not really well known is that structural biology, which is incredibly useful in providing us with information about the structure, doesn't really reach down to the level of detail where the quantum mechanics of a chemical reaction becomes apparent, which is in the sub-Angstrom scale. Most X-ray or NMR structures really are above the 1 or 2 Angstrom scale, and so the finer details of the chemical reaction are not possible to be observed with that. Another thing is that those are static techniques. It's very hard to actually observe the dynamics of an enzyme during the catalysis of that particular chemical reaction, because evidently when you have crystals, that's a static picture, and you require doing large ensemble averaging, and so you're not going to be able to track and observe the chemical reaction at the single enzyme level, which is what we do here. Again, each one of these steps corresponds to the reduction of a pair of sulfur atoms in the backbone of the protein that we've engineered, catalyzed by a single thioredoxin enzyme. Like before, we take many of these events, average them, and we obtain the force dependency of the reaction. You can immediately see that this force dependency is more complicated than what we had before. So here you have that at 25 piconewtons you get a very high rate of reduction of disulfide bonds, but as you increase the force, the rate decreases first and then increases again. This contrasts with the monotonic increase on the rate that we had observed for simple nucleophilic agents such as DTT. So if you plot the rate as a function of force, the rate of reduction as a function of force for thioredoxin enzyme, you see this very striking biphasic shape where the applied force reduces the rate at which the enzyme can catalyze the disulfide bond reduction. And then there is a second phase where, as you increase the mechanical force, the rate of reaction increases again. And we have a simple model that explains this. The model is a modification of Michaelis-Menten type reaction, where the enzyme first has to bind to the substrate, and then, upon binding to the substrate, undergoes sort of a catalytic step that allows the reaction of the disulfide bond. In our model, the binding is independent of the force, but the catalytic step that allows the reaction of the disulfide bond is force dependent. And there is a second pathway, this pathway that you see here, which is completely independent of the first, and it corresponds to a second way of reducing the disulfide bond by the enzyme, and that is the independent pathway number 2 that we call here. And the first pathway, we think, corresponds to a situation where the disulfide bond in the polypeptide binds to a groove in the enzyme, and you can see the groove here is sort of the dark part in the enzyme shown in the background here. The disulfide bond is in the groove, but in order to acquire the correct geometry for the chemical reaction to occur, the disulfide bond has to rotate, and that rotation shortens the polypeptide by an amount that's in the sub-angstrom scale, and it's around 0.7 angstroms. And if you take those numbers and plug them into the simple model, assuming that the force dependency arises from that rotation by the disulfide bond to allow for the catalysis of the reduction, you explain the force dependency quite nicely. Let me jump over all of this. So the first pathway, the pathway that allows for the reaction of the disulfide bond via that force-dependent step that requires the rotation of the disulfide bond, is also something that you can see that's involving the groove, because if you introduce a mutation that hinders the hydrophobic interactions of the groove between the polypeptide and the groove, then you almost completely eliminate that pathway, leaving this second pathway of reduction untouched. I know this is beginning to sound complicated. Most of you are not biochemists, and I was told to use simple words, but there is no escape from talking about an enzyme this way. It's just because of the nature of the machine. But let me say that as a take-home message that you can take from this is that anything that gets shorter will be discouraged by pulling force. And so if the two sulfur atoms have to rotate to align with the catalytic cysteine in order for the disulfide bond to be reduced, that will be discouraged by the mechanical pulling that you're applying to the substrate in this case, right? And if you hinder binding of the substrate to the groove of the enzyme, then that pathway for the catalysis of the disulfide bond disappears, and you can show that with the mutagenesis. When you go from one enzyme type, let's say the thioredoxin enzyme from E. coli that you can see here, this is on scale now to allow the data from the human thioredoxin to come in. You can see the biphasic force behavior of the thioredoxin enzyme from E. coli. And if you take thioredoxin from humans, you see that humans have a much higher rate of reaction of the disulfide bond, a much stronger force dependency, and they don't show this second pathway. And so at this point, we have sort of three different forms of reduction of a disulfide bond that I have shown you using the simple AFM assay. We have the simple exponential increase in the rate that you observe using simple nucleophiles such as DDT. In this particular case, is cysteine. These typically require millimolar concentrations to allow for the reduction of the disulfide bond and have a simple exponential force dependency, as you can see here in this semi-log plot, as a function of force. Then you have the human thioredoxin, which I show you has this negative force dependent in the pathway of reduction, and this negative force dependency is due to the fact that the substrate disulfide bond has to bind into the groove of the enzyme, has to rotate in order to acquire the correct geometry for the SN2 reaction, and that rotation is discouraged by the pulling force. And then there is a third mechanism that you see that the high force rate of reduction of the human thioredoxin does never get to zero. If it was just something that got discouraged by pulling force, you would expect that the higher you pull, you will eventually converge to zero and completely block the rate of reduction of the disulfide bond. But that really doesn't happen. You reach sort of a steady-state level, and that steady-state level becomes force independent, and we think that's just a tunneling reaction where the electrons for the reaction jump without going through a barrier and reduce the disulfide bond by simply allowing the electrons to tunnel through rather than overcome a transition state. All right, so this was an overestimate. Let me just tell you where all of this is going. So we've looked at the chemistry this way, the fine details of the chemistry in terms of the various pathways that depend on force, the rotation of the disulfide bond, or the electron tunneling form, or modes that relate to these simple nucleophiles. And we wonder, how have enzymes that are so prevalent in all of nature change their chemical pathways depends on which branch of the tree of life they belong to. So we've looked at the sort of chemistry details of the force dependency of a number of enzymes from eukaryotic and from bacteria, and all of the bacterial origin enzymes have this two-pathway feature in the force dependency, whereas all of the sort of eukaryotic origin enzymes lack that second pathway of reduction. And we wonder, why would that be the case in terms of recognizing features in the force dependency of these various enzymes? And it turned out that the groove onto which the substrate polypeptide binds in the case of the eukaryotic enzymes is much deeper than in the case of the prokaryotic enzymes. So it appears that through evolution, the groove onto which the substrate polypeptide binds is much, much deeper in the eukaryotes, which are evolved later than the prokaryotes in terms of the development of this particular enzymatic structure. And that seems to be related to the disappearance of this second pathway. So we think that the second pathway in the enzymatic reaction corresponds to something that disappears when the depth of the groove in the enzyme increases, and making the enzyme far more specific. So this is more or less what we've discovered so far. If you look at then a number of enzymes through the tree of life, you can already distinguish from the force dependency of the reduction reaction features in the sub-Angstrom scale which have been inaccessible to techniques such as NMR or X-ray crystallography, and most certainly to test tube biochemistry. Just to tell you, because surely you do not know this, the way that we used to measure the rate of reduction by thioredoxin in a test tube is by reducing insulin. And when insulin gets reduced, insulin precipitates and form clumps and create sort of a scattering fluid. And so if you look at the scattering of light in a test tube where you introduce thioredoxin, the increase in the scattering caused by these clumps of precipitates used to be the way that we measure the activity of thioredoxins. Now, nanocenter, in my mind, always associates with some form of AFM, at least as far as I have seen around the world. You have a bunch of them, for sure. This is biochemistry, enzymology done at the single molecule level using a single AFM. Okay? It's easy to get tangled up in sort of the measurement of the rates and the significance of that if you had no exposure to these questions before, but it's pretty significant that the techniques that you use in the nanocenter can actually capture in the sub-Angstrom scale the dynamics of a single enzyme. Not only that, but also when you go from one enzyme from eukaryotes to another enzyme in prokaryotes and different parts of the evolutionary tree, you see features that changed in the force dependency. And this is totally new. This is something that people haven't really seen before. And so this difference between having the second pathway or not having it sounds funny to you probably because you haven't thought about it yet, but it's sort of a revelation in the sense that you can detect very sensitively these differences in the sub-Angstrom catalytic mechanism in enzymes as they have evolved over time. There is, I'm just going to end with this slide, that there's something pretty interesting that has happened in the last 10 years. And again, I'm thinking in terms of the nanocenter and the contribution that interdisciplinary people such as ourselves are going to make, over the next, in the future, hundreds of years, whatever. And it's that since the first sequencing of the human genome in the year 2000, now there are these machines that can very effectively sequence DNA from just about everything. I mean, there's a number of machines now. 454 is one of them. Everything's going to get sequenced. Absolutely everything's going to get sequenced in the next few years. And the significance of that is profound, because if you align the sequences of, let's say, thioredoxins, which is what I'm showing you here on a very small scale, there are 4,000 thioredoxin sequences from every organism that you can imagine. Computer scientists have developed programs, maximum likelihood programs, where you can actually take these thousands of sequences and then predict what actually led to the extant sequences today. Because we know what the sequences are today from all these organisms. You would like to know, can I reconstruct the complete history of these sequences? It's like reconstructing language. I mean, if you have the sequences, you can actually go back and reconstruct all the sequences that led to the actual sequences today. And these programs are maximum likelihood. They don't tell you with certainty what the evolution of the sequences are, but it tells you what is the most likely history of the sequences all the way up to today. And that's amazing, because it turns out that we have such a reconstruction here from one of my collaborators, Eric Gauscher from the University of Miami. He's a computer scientist. He's taken all of the thioredoxin enzyme sequences that we know of today and created a tree all the way down to the lowest universal common ancestor, which is about 3.5 billion years ago, presumably. And that entire timeline is a timeline made of sequences. So you can take any of these sequences today, and you can actually put them in a plasmid and express them in bacteria. Actually, you don't even need to do that. You can just buy things out. You can buy the primers, send them to a company, and you get back a little tube with an enzyme that maybe existed 3.5 billion years ago. Actually, in my laboratory, my wife has made those enzymes. She's sitting back there. She's the molecular biologist of my group. And so we've taken the sequences, we've made enzymes at various nodes all the way back 3.5 billion years. And amazingly enough, all of the sequences have yielded functioning enzymes that have fine details in the chemistry revealed by the force dependency, and we're actually doing this work right now. And so this interplay between simple instrumentation such as the AFM the tremendous power that we have at nanocenters to manipulate single molecules, the advent of all of the sequence information that we have today, and these profound questions that are raised by biology, I think will make the future extremely exciting for us. Forgive me for having so much detail on the biology and all that, but I think it's interesting that you know that you can combine all these layers together and produce really striking novel information that's unavailable by the standard techniques of structural biology, biochemistry that we have used for the past 100 years. And let me stop here and answer your questions. Why you say that? Well, all the proteins get recycled through the proteasome, the majority of them, or the endosomes in cells. That's correct, but on a very different time scale. Typically the lifetime of most proteins is days or months, whereas the folding and unfolding cycles will be very fast. And so in the actual life of a single protein, you'll probably have to go through cycles of folding and unfolding, or chemistry such as I've shown here, very, very large number of times.
But in your experiment, you didn't expect to see a deterioration of this single molecule, and wouldn't see this folding and unfolding, and you will see that at the end it's like unfolded and that's it, can go back?
Well, you're extrapolating from intuition, and intuition has to do with the fatigue of, let's say, metals, which is totally inappropriate for the case of biological molecules. I mean, in the case of metals, fatigue occurs because of dislocations in the lattice, where you get these motions of the atoms along the planes of the crystal lattice. Let's say, gold is an FCC structure, and so you get these, dislocations as you pull it. That doesn't happen in biology. You have these proteins which spontaneously self-assemble, driven by hydrophobic collapse, where every atom essentially goes back to its original position. So you don't get fatigue in any sort of sense. There's no such thing. You do get chemical changes, though. You might get spurious enzymatic activity, or you might get some ion bridges and other things that happen, but fatigue in the sense of a metal fatigue or anything like that, it just doesn't happen. Right. Okay. So that's an actually very, very good question, because the time scales between simulation and experiment are so fantastically different that the problem is that people like the movies but don't believe the data generally. So the movies I show you are like a couple of nanoseconds. The time scale of the experiments that I've shown you are all in the time scale of seconds. And so you have to sort of take a big leap of faith going from some events that occur in a few nanoseconds. But it's just a matter of computer power, and the computing power is just going to increase steadily until we're able to simulate things on real time. Right now, it's just several orders of magnitude. And if you go to any of the meetings of people considering simulation work, that's the biggest problem, that they don't really know how to scale things appropriately. So the movies are generally right. The numbers they get from the simulations can be more difficult. Surprisingly enough, quantum mechanical simulations, because typically they refer to very few atoms, are far more accurate in terms of quantitative predictions.
This in the microscope?
Well, the PID that I show here has a lag time around 3-5 milliseconds, can be up to 10 milliseconds, but the separation between events is hundreds of milliseconds up to seconds, and so in terms of measuring rates here, the PID does limit the highest rate that you can measure for sure, but it doesn't really interfere with the measurements at this stage. So you have to pull at very high forces to run into the time scale of the PID. And so the ideal instrument is one where the PID is so fast that it doesn't really create any lag in the feedback, and we're slowly going up there. I don't know if you heard of FPGAs, this field array programmable, this field programmable gate arrays that essentially replace all the analog electronics with very fast digital feedback, and we're exploring that to make a very fast feedback into a microsecond timescale. And so that, but it doesn't really affect the measurements, and it doesn't bear any relation to the simulations per se. Yeah.
Some proteins undergo huge conformational change. Can you observe this? And can you calculate the force between a band and extended conformation?
Well, so the proteins I was showing you here, which many of them come from human cardiac titin, are indeed undergoing this fantastic change in conformation. I mean, they go from the folded state, which is about 5 nanometers in length, to the unfolded state, which is about 30 in this particular experiment that I was showing you. So, you know, that's a very large conformational change. Is that happening in vivo? I think it does, because the elasticity of muscle depends on these proteins unfolding and extending elastically. Is this something that we can do without proteins? Maybe is what you're asking in terms of capturing conformational change in other proteins. And every time that you design the experiment, you have to sort of make it AFM friendly or AFM ready in such that the molecules that you're going to be working with will give you some sort of signature for the AFM so that you can recognize it. But I mean, you're thinking the right direction. I mean, the thing I'm trying to excite you about is that you can take a machine, a tool, that's squarely from sort of the engineering, physics realm, and then place it in biology and see things that people have never seen before. I mean, all the stuff I show you here couldn't conceivably be done on a test tube, and it's of great importance for understanding how these proteins work in real live tissues. And I think that excitement can be transferred not just to these projects that I'm doing, but just about any other project that you're doing with nano fabrication facilities and whatnot.
Some proteins fold in parallel. Can you see it? What do you see?
In parallel, what do you mean?
You know, like in masking, not only one ligament would fold and unfold.
Well, they all fold independently. I mean, they're Markovian in the sense that they don't look at the past history and they don't talk to each other. So they're independent agents, and they will all fold following sort of statistical mechanical behavior. So they don't. There's nothing synchronized in their folding, if that's what you're asking. They're all folding independently of each other.
You know, sometimes the force will distribute between two of them, and sometimes if the length is not exactly right, it could fall on one only. Then there should be some interdependence. If you try to unfold two strands of protein.
Yeah, to some extent that may happen, but I think it's small because generally the proteins will be rather soft once they're unfolded. So, but you're right, the force will distribute throughout all of the molecules, and if one molecule becomes shorter, it will bear more of the force than the others that are still relaxed. And so that's a way in which they might communicate with each other. But there are no models of this. You need to take all of the experiments that we've done and other people have done in manipulating single proteins with AFM and then scale them up to some sort of engineering model of the tissues So you really begin to understand where the mechanical properties of all these tissues come from, and that has not been done.
You do those beautiful single protein experiments, but at the end of the day, you average and you get this how observable. And I wonder if there isn't more information in the data. I would have expected that there would be. And if you talk much about other type of observables that you would get with the same measurement aside from the average, like variance,
Right. Yeah, it's true, and it's sort of funny that we go through a world of work to get down to a single molecule level, but then we merely average our data to get rates and such.
well, I
And
wasn't implying that.
no, but that let's start with that. Let's start with that. I mean, it's sort of a funny situation in that that you wonder, well, why don't you just happy with the averages? The ion channel field is sort of my best example, which is really sort of the first field where there was single molecule recording in detail. The ion channel field studied the single proteins in membranes, and it began around 1976 or so. In fact, it began earlier, but Erwin Nair and Bert Sackman invented this patch clamp technique to record from single proteins around 1976. And there were numerous models explaining the electrical activity of, let's say, neurons, which had already made assumptions about what the ion channels would look like. And all those models work extremely well in predicting the shape of the action potential, the duration, all of that. And they were all wrong. That when people look at the first ion channel, they realize that the model on the dynamics of the protein that had been proposed was wrong. It just didn't work that way. And people love writing those papers at the beginning of the 80s saying, revisiting the action potential at the single ion channel level and so on. That didn't change the way we view the action potential. It changed the details. And the way the details mattered is when people start making use of drugs to control ion channels, or let's say that people start isolating ion channels from patients that had particular diseases, it turned out that the details were very important, because if you have a point mutation somewhere, that point mutation has some very specific effect on the activity of the single molecule, which immediately vanishes completely from the average. So the outcome of that effort, which lasted until now, and now you hear less about it because they were so successful that they revolutionized medicine at the single molecule level. So those details became important in designing treatment for particular diseases or improving drugs and so on. But to be able to do that, it takes quite a long march of successful experimentation so that you build a very solid base in which you can approach big questions in biology, and they certainly were very successful. This is just the beginning, and the tantalizing part of this is that we've always assumed that tissue elasticity is just like a rubber band, and nothing's further from the truth. That we know now that it involves mechanical unfolding and refolding of proteins dynamically, and we know that mutations have a big effect on that. I think that it's going to be a while before biology catches up with us, and we begin that conversation that was so profound in the ion channel field. But it will happen. So hang in there. I mean, this is sort of really kind of trivial in the sense that we average the things and we get rates, and we compare rates with other enzymes, and we're happy with the results. But as this gets deeper and deeper into the function of these molecules, I think the details that you see at the single molecule level are going to begin to matter a great deal. One quick example, because my answers are very long, and is that there are many. I used to work at the Mayo Clinic in Rochester, Minnesota, and there are familial forms of cardiomyopathies, of people that have heart muscles that are too soft, So when they fill with blood, they can't contract hard enough to expel the blood at the end. And so it's possible that the mutations are mutations that affect the elasticity of titin. And point mutation where the force at which one of these proteins unfolds is much lower, so the rate increases, and that protein might be unfolding at a much lower force, which will make the tissue more elastic, for example, right? We just don't have such a database of sort of mechanical biology yet, but I'm sure it's there. You know, so it will come, it will happen, and the nanocenters are going to be there, right?
No, I should
So,
say
I
that
think we should stop the formal discussion now. We will be available throughout the conference and at Tel Aviv University for 2 months to come. Thank you again for the very inspiring.
Just one brief point, so you don't think that it's only in Tel Aviv that you have a beach. These are my. Oops, where did it go? I've been disconnected already. Okay. Anyway, you saw the picture. You know, that's the beach in New York.
Are you any of those?
No, I took the photo.